So, good morning everyone. This is Shailendra Parmar presenting a presentation on the topic genetically modified organisms. So, what are GMOs? Genetically modified organisms (GMO) are the organisms whose genome has been engineered in a laboratory in order to favor the expression of desired physiological trait or the generation of the desired biological product. In conventional livestock production, crop farming, and even pet farming, it is a long been practiced to breed select individual of species in order to produce offspring that has a desirable trait. Current use of genetically modified organisms. Agriculture plants are one of the most frequently used side example of genetically modified organism. Some benefit of genetically engineering in agriculture are increase in crop yield, reduced cost for food or drug production, reduced need for pesticides, enhance the nutrient composition and food quality. Other applications include the production of non-protein, bioplastic or non-industrial ornamental plant product. A number of animals has also been genetically engineered to increase the yield and decrease susceptibility to disease. The pharmaceutical industry is another frontier for the use of GMO. In 1986, human growth hormone was first protein pharmaceutical made in plant. Genetically modified crops, GM crops, are the plant used in agriculture, the DNA of which has been engineered using genetically engineering method. Plant genome can be engineered by physical method or by the agrobacterium for the delivery of the sequence hosted in tDNA binary vector. Some examples of genetically modified crops are Bt cotton and pest-resistant plant. Bt cotton. Some strain of Bacillus thuringiensis produce a protein that kills certain insects such as Lepidopteran, Coleopteran and Dipteran. Bacillus thuringiensis form a protein crystal during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Bt protein exits in inactive protoxin, but once insect digest their inactive toxin, it converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activate toxin bind to the surface of midgut epithelial cell and create pores that causes the cell swelling and lysis and eventually causes the death of the insect. Specific Bt toxin gene were isolated from Bacillus thuringiensis, that is insect specific, and incorporated into several crop plants such as cotton. Pest-resistant plant, a nematode Melodic incognitia infect the root of tobacco plant and causes the growth reduction in yield. A novel strategy was adopted to prevent the infestation, which was based on the process of RNA interference. RNA interference takes place in all eukaryotic organisms as well as method of cellular defense. This method involves the silencing of specific mRNA due to complementary double-stranded RNA molecule that bind to and prevent the translation of the mRNA silencing. The source of complementary RNA could be from an infection virus having RNA genome or mobile genetic element, transposomes that replicate via RNA intermediate. Using agrobacterium vector, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in a host cell. These two RNA, being complementary to each other, form a double-stranded that initiated RNAi, thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Transgenic animal. Transgenic animal are the animal with the modified genome. A foreign gene is inserted into a genome of the animal to alter its DNA. The method is done to improve the genetic trait of the targeted animal. Transgenesis is a phenomenon in which the foreign gene with a desired characteristic is introduced into the genome of the targeted animal. The foreign gene that is introduced is known as the transgene, and the animal whose genome is altered is known as transgenic. These genes are passed on the successive generation. The transgenic animals are genetically engineered and also known as genetically modified organisms. The first genetically modified organism was engineered in year of 1980s. Method of creating transgenic animal. Physical transfection. In this method, the gene of interest is directly injected in the pronucleus of fertilized ovum. It is the very first method that proved to be effective in mammals. This method was applicable to wide variety of species. Other methods of physical transfection include particle bombardment, ultrasound, and electroporation. Chemical transfection. One of the chemical methods of gene transfection include transformation. In this method, the targeted DNA is taken up in the presence of calcium phosphate. The DNA calcium phosphate co-participate which facilitate DNA uptake. The mammalian cell possesses the ability to take up the foreign DNA from the cultural medium. Retrovirus mediated gene transfer. To increase the chance of expression, the gene is transferred by the means of vector. Since the retrovirus has the ability to infect the host cell, they are used as a vector to transfer the gene of interest into the targeted genome. Viral vector. Viruses are used to transfer rDNA into the animal cell. Viruses possess the ability to infect the host cell, express well and replicate efficiently. Vectorification. It is a process by which the gene of interest is transferred into the targeted gene with the help of bacteria. Example of transgenic animal. Dolly sheep. Dolly the sheep was the first mammal to be cloned from the adult cell. In this, the other cells from the 6-year-old fine Dorset white sheep were injected into unfertilized egg from the Scottish blackfish egg, which had a nucleus removed. The cell made to fuse by electrical pulses. After the fusion of the nucleus of the cell with the egg, the resultant embryo was cultured for 6 to 7 days. It was implanted into other Scottish blackfish egg, which give birth to transgenic sheep Dolly. Transgenic mice. Transgenic mice are developed by injecting DNA into the oocyte or tooth-celled embryo taken from the female mice. After injecting the DNA, the embryo is implanted into the uterus of the receptive female. Application of transgenic animal. Study of disease. Transgenic animals are specially designed to study the role of genes in the development of certain disease. Moreover, in order to devise a cure for this disease, the transgenic animals are used as a model organism. These transgenic models are used in a research for development of medicine. Biological product. A number of biological products such as medicine, nutritional supplements are obtained from transgenic animals. Research from the manufacturer of medicines to treat the disease such as phenylketonuria and hereditary emphysema is going on. Vaccine safety. Transgenic animals are used as a model organism for testing the safety of the vaccine before they inject it into the human. This is conventionally done on monkey. Advantage of genetically modified organisms. More nutritious food and grow with less use of pesticides. They are faster growing plant and animal. Food with more desirable traits such as potato that produce less of cancer causing substance when fried. Medicinal food that could be used as a vaccine or other medicines. They has a tolerance toward the certain herbicides. The genetically modified organism can be produced by the lower cost. Disadvantages of genetically modified organisms. GM crops disturb the natural process of gene flow because the better trait produced from the engineering gene can result in the favoring of one organism. It is endanger farmer and trade along with the environment. It is more inclined toward the marketization of farming, the work or immortal profit. Genetically modified crop being altered biological may pose a human health risk. The excessive production of GM food with the rendered effectiveness over the time because the pest that this toxin used to dietary eventually might develop a resistance towards them. So thank you everyone for watching the video.